Now we'll discuss about an important topic called as iron metabolism. Iron metabolism. In nature, iron is abundant, but we'll use only little amount of iron, and this iron forms a key constituent of many human proteins. Okay, we'll see which proteins will have iron as its constituent. They are hemoglobin, myoglobin, cytochrome, catalase, or peroxidase. These all compounds are called as heme compounds. Okay. Next, we have non-heme compounds. They are ferritin, hemosiderin, transferrin, iron sulfur clusters. These iron sulfur clusters are seen in biological oxidation. These are called as non-heme compounds. Non-heme compounds. Now we'll see about iron absorption. Iron absorption. This iron will be regulated at the absorption level. So iron absorption is very important. Now we'll see in detail about iron absorption. So it is an highly regulated process. Highly regulated process. This iron absorption will occur in the proximal duodenum. Proximal duodenum. In diet, the iron may be in the form of ferric, that is Fe3 part, or it will be in the form of ferrous part, that is Fe2 plus. This Fe2 plus is easily absorbed. This Fe3 plus cannot be absorbed into the intestinal cells. Cannot be absorbed. So if diet contains more amount of Fe3 plus, then it has to be converted into Fe2 plus. This conversion of Fe3 plus to Fe2 plus can be done with the help of vitamin C, or we have an enzyme in duodenum that helps in conversion of Fe3 plus to Fe2 plus. Now we'll see what happens in the proximal part of the duodenum. Okay, this is intestinal cell, this is the apical part, this is the basolateral part. This is the basolateral part. Now in the apical part, we have a protein called as, which contains an enzyme activity. This is called as duodenal cytochrome B reductase. Okay, duodenal cytochrome B reductase. What it will do? It will convert Fe3 plus to Fe2 plus. So this cytochrome, duodenal cytochrome B reductase will act on Fe3 plus, which will convert Fe3 plus into Fe2 plus. Or directly, if food contains vitamin C, this vitamin C can convert Fe3 plus into Fe2 plus. After that, by formation of Fe2 plus, then this Fe2 plus has to be absorbed into the intestinal cell. For this, we have one more protein, one transporter, that is called as divalent metal ion transporter 1. What is this divalent? It can transfer only the atoms which contain two ions. Okay, divalent, two valency. Okay, that is Fe2 plus can be transported. They can transport manganese. They can transport cobalt. They can transport zinc. They can transport copper. They can transport lead. All these are divalent metal ions. So this divalent metal ion transporter is non-specific. Non-specific. Okay. Then this divalent metal ion transporter will transport iron into the intestinal cell. Intestinal cell. Okay. After transportation of Fe2 ferrous part into the intestinal cell, then it has two fates. One, it will be converted into Fe3 plus and it is stored in a protein called as ferritin. This is ferritin. Okay, iron is stored in ferritin in the form of Fe3 plus, or this iron will pass into this side. We have blood, so this side or towards the basolateral membrane we have a protein called as ferroportin. Okay, portin means it will transport iron from inside to outside. Ferroportin. Okay, it will transport Fe2 plus to outside, and this Fe2 plus is very active form. This Fe2 plus will generate free radicals. How it will generate free radicals? Whenever Fe2 plus reacts with hydrogen peroxide inside the body, it will be converted into Fe3 plus and they will release hydroxyl free radicals. This is called as Fenton reaction. In order to prevent this reaction, this Fe2 plus has to be converted into Fe3 plus. This conversion of Fe2 plus into Fe3 plus, okay, this is done with the help of a protein called as hephaestin. It is done with the help of a protein called as hephaestin. Okay. This hephaestin will contain copper as a coenzyme. This is very much important because if copper is absent, this hephaestin cannot convert Fe2 plus into Fe3 plus. And this Fe3 plus will be taken up by a transporter called as transferrin. This is taken up by a protein called as transferrin. We have transferrin. This transferrin will transport iron in the form of Fe3 plus. This is transferrin. So in diet, if you take iron in the form of Fe3 plus, it has to be converted into Fe2 plus. It can be done with the help of dietary vitamin C, or we have a duodenal enzyme which is called as a duodenal cytochrome B reductase, which will convert Fe3 plus into Fe2 plus, and only Fe2 plus will be passed into the intestinal cell with the help of a divalent metal ion transporter 1, and this Fe2 plus is converted into Fe3 plus and stored in intestinal cell in the form of ferritin. Later it is transported outside with the help of a ferroportin, and this Fe2 plus again has to be converted into Fe3 plus, and this Fe3 plus is transported with the help of a transferrin protein. The conversion of Fe2 plus to Fe3 plus is done with the help of a hephaestin which contain copper. Next, transferrin carries 300 micrograms of iron per deciliter, and that is 300 micrograms of iron can be bound to the transferrin. This is called as TIBC, that is total iron binding capacity. And that means 30% of transferrin is occupied by iron, that is 30% iron binding sites are occupied by iron. This TIBC is decreased to 16% in iron deficiency anemia, and it is increased to 45% in iron overload conditions. This transferrin is a glycoprotein. Where carbohydrate requirement is must. Whenever this carbohydrate can be decreased in quantities in alcoholics, and in alcoholics there will be carbohydrate deficient transferrin. So in these alcoholics there is defect in transport of iron. Next we'll see about ferritin. This ferritin helps in storage of iron. This will be around in the range of 50 to 200 micrograms per deciliter in plasma. And this ferritin will occupy 3000 to 4500 ferric atoms. So this ferritin will be an indicator of iron stores, because iron is stored in the form of ferritin. Now this ferritin is degraded. If it is partly degraded, then it is called as hemosiderin. This hemosiderin, collectively called as hemosiderosis, this hemosiderosis will contain lot of ferritin molecules, but release of iron is very less in hemosiderosis. Hemosiderosis contains lot of degraded ferritin molecules, but they will release less iron. This is about ferritin. Now, after iron is taken up by transferrin, then it has to be transported to the tissues. Then what will happen at the tissue level? We call that cycle as a transferrin cycle. What happens in the transferrin cycle? We have a receptors on the cell membrane. What are those receptors called as? TFR1. What is TFR1? That is transferrin 1 receptors. Okay, these transferrin receptors will take up transferrin. This transferrin will contain iron in the form of Fe3+. Iron in the form of Fe3+. Okay. Next, these receptors are coated by a specialized protein called as clathrin. This is called as clathrin. That means iron is absorbed in the form of receptor mediated endocytosis. This clathrin will help in receptor mediated endocytosis. This transferrin is called as holotransferrin. Whenever iron is occupied in the transferrin, then it is called as an holotransferrin. This holotransferrin will bind with the transferrin 1 receptors which are attached to the clathrin coat, and there will be an endocytosis of the receptor along with the transferrin endocytosis, and they will contain clathrin coat. This is called as an early endosome. They will contain transferrin molecule with Fe3+, and this is called as early endosome. Outside pH is 7, and gradually this early endosome is converted into late endosome. After this clathrin coat reaches to the cell membrane, and it is ready to bind with the transferrin receptors, and this early endosome is converted into late endosome. What happens in the late endosome? The pH will be decreased to 5. pH is decreased to 5, and this transferrin will release iron molecule, and this Fe3 has to be converted into Fe2+. This is done with the help of ferrireductase. And this converted Fe2+ is transported again back to outside with the help of a protein called as ferroportin. Not ferroportin. It is done with the help of divalent metal ion transporter 1, DMT1. And this iron will be utilized for metabolism. And this transferrin without iron is called as apotransferrin. Without iron it is called as apotransferrin. And this apotransferrin, we have receptors also here. and this apotransferrin and the TFR1 receptors are transported back to cell membrane. Transported back to cell membrane, and this apotransferrin goes back to the intestinal cell, and transferrin receptors will wait on the cell membrane to accept one more holotransferrin. This is called as transferrin cycle. As we have TFR1, that is transferrin 1 receptors, we also have TFR2 receptors. These receptors are found in liver. They act as a sensors for iron. That is about transferrin cycle. And this is regarding iron absorption. But inside our body, iron is also released from RBC. Normally, RBC lifespan is 120 days. After 120 days is completed, then this iron is released from RBC. These detected RBCs are phagocytosed by macrophages. And these macrophages will digest RBC. This RBC will release iron. This iron has to be converted into Fe3 plus. Why into Fe3 plus? Because this Fe2 plus is very reactive and it cannot be transported directly. So it has to be converted into Fe3 plus. This is done with the help of a protein called as ceruloplasmin. This is a copper transferring protein which is very helpful in the conversion of Fe2 plus to Fe3 plus. So at the intestinal level, we have hephaestin, which contains copper, which helps in conversion of Fe2 plus to Fe3 plus. And this ceruloplasmin also helps in conversion of Fe2 plus to Fe3 plus. This is about what happens whenever iron is released from the that is, iron is released from the RBC, where RBC is digested by macrophages, and they will release Fe3 plus. And this Fe3 plus is taken up by transferrin and it is transported to the tissues. So this is about iron absorption and transport system. Now this iron has to be regulated tightly. Okay, we have two mechanisms. One is at the intestinal level, another is at the gene level. So at the intestinal level, we'll call it as a mucosal block theory. What happens in the mucosal block theory? This is an intestinal cell, and this is a macrophage. After absorption of iron from intestinal lumen into the intestinal cell, then it is transported by a protein called as ferroportin. Okay, we have seen that it is transported by ferroportin. And from macrophages also, this iron is transported with the help of a ferroportin. Okay, then what will happen? Liver will synthesize a protein called as hepcidin. Whenever iron is increased, then liver will secrete an protein called as hepcidin. What this hepcidin? will do, this hepcidin will go and bind with the ferritin. Go and bind with the ferritin. What will happen? This binding of hepcidin with the ferroportin, they will cause internalization. That is, ferroportin along with hepcidin, they will be internalized. That is, internalization of ferroportin and hepcidin, and they cause degradation of ferroportin. And same thing will happen in macrophages also. Ferroportin is internalized and they will be degraded. Both things will happen in the intestinal mucosal cell and this macrophage. Okay. And what will happen? Whatever iron is absorbed into the intestinal cell will stay inside the cell only. This will, because this ferroportin is not there, whatever Fe2 plus is absorbed, it will stay inside the cell only. Whatever Fe2 plus which is released from the RBC, it will be staying inside the macrophage only. That means there is a block that is called mucosal block. So if there is a mucosal block, this will cause decreased iron absorption. And this will cause decreased recycling of iron liberated by RBC. What will happen ultimately? They will decrease circulating iron levels. Whenever iron levels are increased, this process will happen. Whenever iron increases, it increases hepcidin levels. This is called as a mucosal block theory. What happens to the intestinal cell? Gradually, intestinal cells are sloughed off. The iron will also go away with the intestinal cells. That is iron regulation by mucosal block theory. And iron is also regulated at the gene level. What happens at the gene level? We'll see. Synthesis of transferrin receptor 1 and ferritin are reciprocally regulated. What will happen here? We have two regulation points. One is TFR1 receptor site and one is at ferritin. Okay. We have iron response element and iron regulatory protein. These two proteins are required. What happens at the gene level? In a mRNA, we have iron response element. This is 5' end, 3' end. Towards the 5' end, we have iron response element, and IRP is there. This IRP will have iron response element binding site. And on the transferrin receptor side also, the mRNA, on the 3' end, we have two iron response elements. This is 5' end, this is 3' end. These are iron response elements, and we have IRP. If iron is increased, what will happen? This iron will bind with the IRE binding site on the IRP. If this IRP is occupied by the iron, then IRP cannot bind. This IRP cannot bind with IRE whenever iron levels are increased. Iron levels are increased. What will happen here? Whenever IRP is not binding with the IRE, this mRNA will be translated and we get a protein called as ferritin, which helps in storage of iron in high iron conditions. What happens at the TFR1 receptor site? Whenever iron is increased, IRP does not bind to IRE. Same thing, if increased iron levels causes binding of iron to the IRP at the IRE site, and it prevents binding of IRP to IRE on the transferrin receptor site. Okay, transferrin receptor 1 IRE response, iron response element. If that is not binding, then this will be degraded and no transferrin 1 receptors are synthesized. If no transferrin 1 receptors are synthesized, then what will happen? Whatever iron is there, it will not be absorbed into the cells. At the tissue level, there is no absorption of iron into the cells, so there is no damage to the tissues. This is what happens at the increased iron levels. What happens at the decreased iron levels? Then at the ferritin site at the 5' end, then iron regulatory protein, this is iron regulatory protein, will bind with the iron response element. This is 5' end, 3' end. What happens at the transferrin receptor level? Here also this IRP will bind with the iron response element. This is IRP, this is IRP, this is iron response element. Whenever iron is decreased, this condition will happen. That means there will be blockage of translation, and no ferritin is synthesized, What happens here? There will be translation, and TFR1 is synthesized. Whenever iron levels are low, you require more amount of transferrin 1 receptors so that iron will be taken up inside. Then you, in low level iron conditions, you does not require ferritin, because storage is not required in low iron conditions. Whatever iron is present, that has to be supplied to the tissues, so that normal hemoglobin, normal myoglobin, every tissue is occupied with the iron. So for that process to occur normally, your transferrin 1 receptors levels have to be increased. More transferrin 1 receptors are required, so that whatever iron is present in the body, that will be taken up by the tissue cells. Okay, this is about regulation of iron metabolism. This is very important. Okay, iron regulation, mucosal block theory. It is a common question which is asked in for UG exams and for PG questions also. And coming to factors affecting iron absorption. What happens here? Factors which will increase absorption are gastric acid, ascorbic acid, sugars, amino acids. All these will increase iron absorption. iron absorption. The why in this iron absorption is increased? They increase the soluble nature, soluble nature, and it increases absorption. But oxalates, phytates, phosphates, these are present in vegetables. These phosphates are present in milk and egg. Tannates, these are present in tea. All these will form insoluble complexes with iron, and they will decrease iron absorption. This is about iron absorption and transport and regulation of iron. This ends the iron metabolism.